Good morning students. Today is lecture 19 of module 7 and in today's lecture we are starting actually ultrafiltration. So, we will learn the basic principles of ultrafiltration, the various advantages of ultrafiltration, the membranes and modules that is designed for the ultrafiltration and different types of ultrafiltration configurations, means the different way how ultrafiltration can be carried away. So, as you know it is a pressure driven process in which water and low molecular weight solutes permeate through the membrane, while the particles, colloids and other macromolecules are retained or rejected. So, the primary mechanism of ultrafiltration is size exclusion. That means basically it is size based separation, although electrical charge and surface chemistry of particle or membrane may affect the efficiency also. Ultrafiltration has developed since 1960s as a consequence of the development of the asymmetric RO membrane. However, unlike RO which is basically or mainly deals with desalination and similar allied operations, ultrafiltration has a wide range of applications. We will see in subsequent discussions in our lecture what are the different types of ultrafiltration applications. So, ultrafiltration pore ratings ranges from approximately 1000 to 500,000 Dalton, thereby making ultrafiltration more permeable than nanofiltration. So, this is one of the most important thing. Since only high molecular weight species are removed by ultrafiltration, the osmotic pressure difference across the membrane surface is negligible. So, we are not considering the osmotic pressure difference actually in ultrafiltration because only high molecular weight species are getting removed, not in the case of RO. Okay. Therefore, in ultrafiltration the applied pressure is required is actually lower than RO to achieve high flux rates, and ultrafiltration can be used to accomplish one or more of the following objectives. So, either you can target a concentration of solutes thereby removing the solvent, or you can have a purification of solvent by removal of solute, and fractionation of solute. So, we have binary fractionation, we have tertiary fractionation. Okay. So, analysis of complex solutions for specific solutes. So, you can see this particular scheme is telling us about the first one. Okay. It is telling us about what the ultrafiltration membranes are doing basically. You can see water and monovalent salts, acids, caustic as all along with some of the amino acids and sugars, they are permeating through the membrane because they are this one cut off, or the size is less than that of the pores. Please remember that ultrafiltration is a porous membrane, though it is asymmetric, but it has pores. So, mostly ultrafiltration membranes are skin membranes. Okay. So, this you can consider as a support, microporous support. Okay. Right, and over that there is a skin layer. Okay. It is just like your RO, okay, asymmetric membranes, but RO membranes are not actually porous, but here the skin layer. Okay. So, this is also porous, right. So, that is why you need to understand this. Okay. Due to the nature of the ultrafiltration membrane, in spite of having it is asymmetric in nature, it will permeate some of the solutes along with the solvent, which is essentially water. Okay. However, it will reject viruses, proteins and different macromolecules, bacteria, fats and enzymes and suspended solids. So, most of the times ultrafiltration also used in water treatment to remove the TSS, the total suspended solids. So, here you can see in another example where ultrafiltration what it is doing is been classified into three things. First is this minimal reduction, then partial reduction or complete or significant reduction. So, this reduction means it is rejection. Okay. So, you can see algae is partial rejection because there are algae, there are microalgae, cyanobacteria and all these things. Arsenic, okay, one of the most this one talked about heavy metal, okay, because of its pollution that it creates in so many parts of the world, right. So, its reduction is also minimum, whereas your bacteria, benzene, then pesticides, okay, then viruses, they are completely rejected. Then some of other heavy metals like lead, mercury, copper, chloride, all these things, they are partially rejected. So, this particular slide makes us understand that what can be achieved or what type of rejections can be achieved for what type of solutes, okay, using ultrafiltration membranes. So, in conventional ultrafiltration configurations, the process solution is usually pressurized, typically between 10 to 70 psi, while in supported semi permeable membrane. Now, solutes smaller than the molecular weight cutoff of the membrane, they will emerge as the ultrafiltrate. Okay. Retained molecules are concentrated on the pressurized side of the membrane. So, usual scheme of operation for any membrane is something like this. Okay. So, you need to pressurize the module, right. So, you can do it by many ways. So, either you can have a pump or you can have a compressed gas, as for example, nitrogen. Okay. So, your feed is coming here, right, entering, and then you get your concentrated solution retentate, and in permeate you are getting the ultra filtrate. So, ultrafiltration is used to separate solids range from 5 kilodalton to 500 kilodalton. So, this is the solute size that can be separated using, or you can say the rejected using the ultrafiltration membranes. So, to achieve this, what should be the pore diameter or pore sizes of the ultrafiltration? So, it is almost 1 to 20 nanometer that is required to have a ultrafiltration membrane to reject 5 kilodalton to 500 kilodalton solutes. So, please note that the demarcation of ultrafiltration and other processes, membrane processes such as reverse osmosis and microfiltration is not often very distinct, but it is all the size of solutes that they are separating, and of course, the type of membrane as well as the pressure range that is required to do the separation. So, ultrafiltrations, one of the most successful use is concentrate macromolecules. So, however, it is also used to fractionate macromolecules that has gained a lot of attention since last few years, right. Initially, it was when ultrafiltration came into picture. So, the initial applications were protein concentration. Okay. Later on, the fractionation of macromolecules also been achieved using ultrafiltration. So, the view that ultrafiltration is a completely size exclusion process is only first approximation. As you know that there are many other factors that play a role in the separation of anything, okay, any membrane based separation. So, they are solute membrane interaction, solute interaction, mode of solute transport. Especially in ultrafiltration, the mode of solute transport can be either diffusive or convective, or can be a combination of both. The first commercial use of ultrafiltration was for the this was what I told you just, for the concentration of proteins. So, ultrafiltration based separation and purification is attractive because that the throughput of the product is very high, the process cost is actually low, and the scale up is very easy. So, the major areas of applications are listed here. They are fractionation of macromolecules, removal of solvent from solutions of macromolecules, diafiltration. So, diafiltration is a classic example or a mode of operation of ultrafiltration. I will show you the scheme later on. So, here what is happening, the removal of the salts are actually carried out and other low molecular weight compounds. Then the removal of cells and cell debris from the fermentation broth, virus removal from therapeutic products, harvesting of biomass, that basically cells, mature cells, membrane reactors and bioreactors, and effluent treatment, both for domestic as well as industrial. What should be a ideal ultrafiltration membrane look like? So, what type of features they should have? So, ultrafiltration membrane should possess high hydraulic permeability to solvent. They should have a sharp retention cutoff. We have already discussed what is this retention cutoff, sharp and diffusive. It should have a good mechanical durability. The mechanical strength should be good, good chemical as well as thermal stability for various types of applications, excellent manufacturing reproducibility as well as the ease of manufacture. Reproducibility is one of the most important factor when you talk about commercial membrane manufacture. So, UF membranes usually differ from the microfiltration membrane in terms of their morphology. So, microfiltration membranes are symmetric membranes, pore shape and size may be different. However, ultrafiltration are asymmetric. I just draw you and show you how there is a skin layer, right. So, microfiltration membranes do not have a skin layer. Okay. So, however, ultrafiltration layer do possess a skin layer, but it is a porous skin layer. So, do not confuse with the RO membrane which is also a asymmetric membrane. Okay. So, this skin layer gives the selectivity to the ultrafiltration membrane, whereas the microporous layer is providing the support, mechanical support basically. The thickness of the skin layer usually ranges from 0.2 to 10 micron depending upon the material and also the intended application. But again you remember, just like even RO also, here also the thickness of the top layer, that is the skin layer, must be as thin as possible. Okay. So, because if it is more thick, then it will provide an extra membrane resistance to the overall resistance of the flow of the solvent or solute. So, which is not good. So, let us understand the different advantages of the ultrafiltration processes. So, for industrial application, ultrafiltration processes offers the following advantages. The first one is that the process can be carried out at ambient temperature, right. So, thus avoiding thermal as well as oxidative degradation of products. So, no thermal requirement is there here. However, in such cases solute diffusion coefficient is temperature dependent. So, second thing is that ultrafiltration permits the removal of water up to 90% at ambient temperature. Since there is no phase change, occurring. So, breaking of emulsions, collapse of gels and mechanical damage do not occur. No solvent or other precipitating agents are required for the concentrating purposes. No generation of wastewater. Please, this is one of the thing which you can compare with RO, reverse osmosis. There is a RO reject, right. So, here there is no generation of wastewater. So, keeps essential minerals in water unlike your RO, and can be installed quickly and fitted easily. So, these are some of the basic informations of ultrafiltration processes. So, the driving force is of course the pressure gradient, usually 1 to 10 bar. The transport mechanism is pore flow. Separation principle is actually sieving, or you can call it size exclusion. The size of the retained species or the rejected species lies between 100 to 10 to 100 angstrom. Membrane type can be asymmetric, porous or composite. Membrane material can be polysulfone, polyether sulfone, polyvinylidene fluoride, polyacrylonitrile, polyamide, aliphatic polyamide, cellulosic, and as well as ceramic also. Ceramic ultrafiltration membranes, mostly tubular type, is becoming very popular nowadays for most of the commercial applications. Pore size is usually 1 to 100 nanometer, flux is 50 to 1000 liter per meter square hour, and energy consumption is 10 to 150 watt per meters cube. So, the important characteristics of the membrane material are porosity, morphology, surface properties, mechanical strength as well as chemical resistance. So, this we have just seen what are the different types of materials, membrane materials that can be used to prepare a ultrafiltration membrane. These membranes usually have two layers, a thin layer which is 0.1 to 0.5 micron layer made up of cellulose ester, and a substructure support material, the more microporous one. So, most of the today's membrane are prepared by a slight variations of the Loewen-Sohry-Rajan process. During manufacturing of the ultrafiltration membranes, their membranes are casted onto the membrane support. So, only the semi-permeable thin layer is in contact with the sample during the process, right. So, the support material below the membrane does not affect any filtration characteristics. Ultrafiltration membranes are composed of a polymer such as polysulfone or polyamide, which is usually extruded into flat sheets or hollow fiber or cut into discs as required by the specific applications. So, when you are making a ultrafiltration polymeric membranes, it is very easy to do that. So, you can make into various shapes, whether you can make into hollow fibers, right, you can make into flat sheet membranes, you can make spiral wound membranes, but when you talk about a ceramic ultrafiltration membranes, you cannot have different types of shape and sizes, though there are plate and frame ceramic membranes are available, and most of those are actually tubular, right. So, plate and frame has a lot of problem, but as you know that ceramic membranes are usually they are brittle, and since the sizes will be and they are very heavy. So, carrying and transportation is a big problem. If there is a big plate type of membrane, okay, the fixing it has to be done it very carefully. Little pressure on that will actually damage the membrane, and the entire membrane, flat membrane can become can break. So, this is why most of the ceramic membranes, ultrafiltration membranes are actually they are tubular. So, a small disc of ultrafiltration membrane may be subjected to rapid fouling and produce a low flow rate for many processes. As a result, UF membranes are typically arranged in a configuration to maximize surface area and reduces fouling by using a tangential flow design to reduce solute accumulation at the membrane surface. That means, most of the times the ultrafiltration membranes does not matter what type of module it is fixed in a cascading systems or in series. We'll today only we'll see how we go actually carry out multi-stage operations. Okay. So, tangential flow ultrafiltration devices may be spiral wound cartridges containing several square feet of membrane wrapped into a central core tube, or a hollow fiber cartridges containing dozens of thin film UF membrane fibers. Now, since fouling is a major problem in ultrafiltration, membranes which are hydrophilic are usually used because they foul less. So, for high purity water application, membrane module materials must be compatible with the chemicals such as hydrogen peroxide used in sanitizing the membrane on the periodic basis. Inorganic membrane materials such as ceramic, carbon based membranes, zirconia etc. have also been commercialized by several vendors for ultrafiltration applications. But we just discussed that what are the disadvantages of having ceramic membranes, but there are plenty of advantages also. Membrane surface charge also plays a very important role during the separation. If the membrane has a slight negative charge, then adhesion of colloidal gel layer to the membrane, okay, is reduced, thereby achieving a higher flux and reducing fouling, subsequent fouling. But in case of slight positive charge, the effect is reverse. That means that concentration polarization build up will be faster, and since it is a pressure driven process, some of these solutes will try to squeeze themselves inside the pores of the ultrafiltration membrane and thereby fouling the pores as well as the membrane. So, charge as well as hydrophilic character of the ultrafiltration membrane comes from the chemical structure of the membrane material and can be a result of chemical grafting or surface treatment. Of course, that depends on the intended application. What is your intended application or targeted application? What you want to achieve? Are you going a separation? Are you going for a concentration? You are going for a purification? You are going for fractionation? There are so many things can be done in ultrafiltration system. So, depending upon that intended application, you can tailor make a particular ultrafiltration membrane as well as module. Now, let us understand the different types of modules. We have been discussing membrane modules in general, then we have discussed the membrane modules in detail in the RO systems. Here we will just in a brief we will discuss, because most of the modules configurations are same, however, their operating features are different. So, instead of understanding the module and their operation today, we will try to understand what are the operating systems or operating parameters for these particular modules, what can be done, what cannot be done with respect to ultrafiltration. So, usually two types of modules exist for ultrafiltration systems. The first one is constant pressure, but a variable flux systems, and the second one is constant flux and a variable pressure systems. Now, what are those? Now, constant pressure and variable flux systems are very popular till 1990s. Later on, what happened actually, the constant flux and variable pressure systems has come into picture in 1995. Okay. They are mostly popular for the submerged system. So, submerged membrane reactor, submerged membrane bioreactor, you might have heard sometimes. We will see what are those. Okay. So, in case of constant pressure and variable flux systems, fouling are controlled by the rapid circulation of the feed solution across the membrane surface. So, these are cross flow operations. So, constant pressure and variable flux modules operates in a cross flow system. So, in constant flux and variable pressure systems, fouling is controlled by continuous air spurging as well as regular back flushing. They are mostly actually submerged systems. So, let us now understand the constant pressure and variable flux ultrafiltration membranes, or per se we can call them cross flow ultrafiltration membrane systems. So, 4 basic modules are available: tubular, plate and frame, hollow fiber and spiral wound. So, now I am not going to discuss in detail about all these things, as we have discussed this earlier also. They are typical configurations, right. Now, but we will try to understand with respect to ultrafiltration how they are. So, the first commercial ultrafiltration system were based on tubular and plate and frame modules. Later on, due to lower cost, spiral wound actually and as well as capillary hollow fiber modules gained popularity. However, still for high fouling solutions, tubular and plate and frame modules are used. spiral wound is usually preferred for a less non-fouling or a very less fouling applications. Yeah, these are hollow fibers. This is a hollow fiber module, ultrafiltration module. This is your tubular ultrafiltration modules. Okay, and this is plate and frame ultrafiltration modules. Okay. So, due to large diameter, tubular ultrafiltration modules can be used to treat high fouling solutions. In applications such as treatment of electrocoat paint, concentrations of latex solutions or separation of oil water emulsions, the fouling resistance and ease of cleaning of tubular modules outweighs their cost, large footprint and high energy consumption. So, basically there will be question, there will be thoughts coming into mind that what type of module, what type of membrane has to be used. As I told you earlier many times, I am again repeating that the intended application will tell us what type of membrane as well as module we are going to use. Let us say for one example, I have to remove virus from therapeutic products, okay, for the pharmaceutical industries, in the biopharmaceutical industries. Okay. So, that is for human consumption. So, you can understand that all the viruses any such nuisance which is present in the initial feed solutions needs to be removed. So, in that when we are designing such a system designing a membrane system, may it be ultrafiltration or anything, then for the therapeutic applications, then please note that we do not think about cost that much. Of course, cost is a factor, anyway it is for the therapeutic applications needs to be removed. Now, let us say we are talking about a wastewater application where we are removing the different components of the wastewater stream, okay, and we want almost a water in which the pollutants, it should be free from pollutants as well as microorganisms. So, here we can little do compromise depending upon the what is the intended application of the water which is coming from the membrane system. Are you going to use it as a potable water? If it is a potable water, then maybe RO is fine. It may needs little remineralization, but that is a different issue, and if it is not so, then ultrafiltration also give us ultra pure water, okay, may not be directly used for the drinking water purposes, but can be used for other purposes, right. So, you can have microfiltration membrane also. So, cost is we have to see actually what is the intended application, then you decide what type of membrane as well as module has to be considered. So, in case of tubular ultrafiltration membranes, usually several 5 to 8 feet long tubes are manifolded in series. So, the feed solution is recirculated through the module at velocities of 2 to 6 meter per second. You can see that the cross flow velocity is very high actually, okay, 2 to 6 meter per second. So, due to this high velocity, the pressure drop of 2 to 3 psi per tube or 10 to 3 psi per bank occurs. The bank is something which is also, you can, I am just trying to draw one, you can see I can call this is a tube bank, okay. So, this is a bank, okay, and this is a single tube, okay. So, due to the high flow rate of the cross flow rate, so the pressure drop is almost 10 to 30 psi per bank. Moreover, for this high circulation rate and pressure drop, large pumps are needed. So, that means you are adding more cost of the energy. So, tubular UF plants often use 10 to 15 kilowatt per hour energy per cubic meter of permeate that is produced. So, the next is plate and frame units. So, they compete with tubular units in some applications. They are less fouling resistant compared to tubular ones, but are relatively less costly. So, when that is what I was telling you, if you are going for a application which are not therapeutic product application, you can always go for plate and plate system or some other membrane applications. Okay. Right, mostly it consists of a flat membrane envelope with a rubber gasket around its center outer edge. Now, these envelopes along with spacers forms a plate that is contained in a stack of 20 to 30 plates. So, one single plate I am trying to show something like this. So, it is a single plate. So, you can have so many different types of plates, okay. So, and thereby making it a stacks of plates. So, you can have in a stack, right. So, usually a stack contains 20 to 30 plates. Typical feed channel heights are 0.5 to 1 mm and the system can operate under high shear conditions. So, they are especially good for bioseparation application. Bioseparation usually means that separations of proteins and antibodies. Okay. So, these units have less lifetime, however, the high value separation that they perform as for example, the bioseparation, okay, they can support this cost. they can be operated at high pressure than tubular and capillary modules. Usually 150 psi is not very uncommon. Their compact design, then small hold of volume and absence of stagnant areas make them suitable for sterilization, right. So, when you are doing bioseparation applications, that means you are purifying say or concentrating proteins, then sterilization of the modules as well as membranes is a must. So, they are sterilizable. This is one of the beauty of this plate and frame systems. So, for these reasons plate and frame units are used in pharmaceutical and food processing industries. So, the next category is hollow fiber modules. So, they are introduced in 1970s by Romicon. So, typical hollow fiber module contains usually 500 to 2000 fibers, single fibers, okay, with a diameter of 0.5 to 1 mm, housed in a 30-inch long and 3-inch diameter cartridge. Now, please understand that this is not something sacrosanct. So, this is usual, okay. It can be smaller than this size or it can be larger than this size also. So, the membrane area is usually about 2 to 10 meter square. So, feed is pumped down the bore of the fibers and operating pressure are quite low, normally not more than 2 bar. The normal feed to residue pressure drop for a capillary hollow fiber module is 0.2 to 0.5 bar. Now, with this they achieve good throughputs with many solutions, ease of cleaning along with small hold of volume and clean flow path. They are used in biotechnological applications. So, the next one is spiral wound modules. So, spiral wound is adapted for UF systems, though initially developed they are developed for the reverse osmosis systems. So, unlike RO, here in case of UF the spiral wound modules, they have coarser feed spacer material, okay, often as much as 45 mil thick. So, 1 mil is equals to 0.001 inch. Okay. So, this unit is usually used when we are talking about this spacer materials. So, these coarse spacers prevent particulates from lodging in the spacer corners. Okay. To have a long term use, pre-filtration is of feed down to 5 to 10 microns is usually required, so that the clogging will be less, and they are especially used to produce ultra pure water for electronics as well as pharmaceutical industries. The development of these modules actually began at around 1995. So, we are talking about a constant flux. So, we discussed about constant pressure and variable flux models. Now, we are discussing constant flux and variable pressure systems. Now, the classic example is the submerged membrane modules, in which a constant flux is maintained by a variable speed pump sucking the permeate through the membrane. Now, fouling is controlled by either air spurging as well as regular back flushing. So, in 1980s, Kazuo Yamamoto and his co-workers developed submerged hollow fiber membrane modules for sewage treatment. Now, initially these units consist of a looped hanks of hollow fibers immersed in a substrate solutions. Now, fouling is very severe. However, they have showed that regular air spurging, with regular air spurging, fouling can be checked or it can be minimized. Two most common types of these modules are submerged hollow fiber, that is capillary modules, or submerged membrane plates. So, in the next one I am showing you the diagrams actually. So, these are submerged hollow fiber UF membrane modules, and this is plates. Okay. So, you can see here how actually the hollow fiber the plate system looks like. You can see this air and water both are flowing in this direction, okay, and we are getting here aeration is done inside this. This is the membrane, this is another membrane. So, aeration is done here in between the space between this membrane, and this is this yellowish color you can see, this is the supporting panel. Okay. So, you are getting the permeated water from here, okay, from the opposite side, okay, and you will see the sludge deposition on the surface of the membranes, okay, and eventually they will be taken out, and this is a two membrane system this particular image is showing. However, you can see here there are so many membrane plates are stacked together, maybe more than 50. Okay. Now, let us try and understand one of the most important thing, that is the common modes of ultrafiltration operation. Okay. So, there are various modes. So, most commonly these are the modes of operation: total recycle operation, batch operation, diafiltration, single pass operation, feed and bleed operation, and multi-stage operation. So, I will try to draw the images, okay, or the sketches. So, let us see how the total recycle operation actually looks like. So, initially you will have a feed tank, of course, you will have a feed tank everywhere, right. Then feed is pumped. So, pumped to a membrane. So, I am showing the cross flow operation, we are talking about cross flow operation, right. Then since we are talking about the total recycle mode, so right. So, this is your feed, right. This is your retentate, okay, which is completely recycled, and whatever its permeate also completely recycle, right. So, this is your feed tank. Now, you know how why this is actually required. So, in this total recycle mode operation, because most of the times when you are trying to develop a membrane and you want to characterize it, study its properties and its performance, initially we will study the membrane using this total recycle mode. Okay. However, they are not very popular in commercial and industrial applications. In the lab scales they are carried out, right. The second one is the classical batch operation. Many times you will see many books they are writing that batch concentration, they are the same, right. So, you have a feed tank, okay. Then your feed is getting pumped to a cross flow membrane, right. So, you will get your permeate here, and your retentate you will be taking and back to the membrane, to the feed tank, right. So, there is a pump is of course required to feed the this one feed solution to the membrane. Now, batch concentration is carried out under a constant transmembrane pressure, right, TMP. Okay. So, however, your flux will decline as the time increases. So, your flux decline profile will be like this. Okay. So, if it is Jv by T time, okay. So, you will see the instant flux decline profile, right. So, let us understand the next one. So, next is diafiltration. So, as I told you previously, so the diafiltration is actually carried out to remove salts as well as low molecular weight solutes. Okay. So, let us see how this looks like. Again the feed tank, there is a pump that is feeding to the cross flow membrane system. Okay. So, in the diafiltration, so the retentate is getting circulated, okay, and we get the permeate here. Okay. So, what is happening actually, we can separate solutes also here, two solutes. However, what is happening in diafiltration is that the solvent, okay, is getting reduced. So, solvent is getting reduced here, right. So, we need to add buffer. So, we add actually buffer here. Okay. So, it is called diafiltration buffer. Usually it is nothing but a solvent only, right. So, separation of two solutes is usually carried out using diafiltration or to remove or reject low molecular weight compounds. So, since the solvent is getting lost, so solvent needs to be replenished. So, using a buffer. Now, single pass operation. Now, single pass operations they are also same thing. It is a feed tank. Okay. So, there is a pump which is getting feed to the cross flow system. So, it is retentate and you get it permeate. Now, this is a typical single pass system. Now, what is happening in this? Now, try to understand that in a single pass system we cannot achieve the targeted separation, and they are not common also in industrial parlance, because they requires very high feed flow rates. Okay. So, single pass cannot achieve the desired separation. That is why there are other things has come into picture. For example, feed and bleed operation. So, let us now understand what is feed and bleed operation. So, I will draw a single feed and bleed system. Okay. So, there is a difference of pump. This pump is called recirculation pump. Okay. In case of feed and bleed operation, this is called a recirculation pump. Okay. So, you are seeing what? This is the retentate. Okay. So, this is called recirculation pump. So, what is happening here actually? High fluid solution velocity. Okay, we can maintain high feed flow rate basically. Okay, the part of the retentate is being fed back to the loop just before the recirculation pump. Okay. So, that means what is the major thing it is happening here? Volume of product that I am taking out, right. So, that is this much, and the volume of the retentate that is being again recirculated here are the same. It is actually called bleed out, that is why feeding and bleeding continuously is taking. So, feeding continuously is happening here and bleeding continuously is happening here. So, this is a continuous process. So, flow rate of recirculation loop is almost this loop is almost 5 to 10 times okay, that of feed flow rate. Okay, feed flow rate. Now, we can have multiple feed and bleed units that are joined in series or in cascades, right. So, now let us try to understand the multi-stage operation. Now, multi-stage operations has come into picture because you know in a single unit operation system, any process, whether it is adsorption, absorption, distillation, anything, in a single stage system the efficiency is usually very less, restricted to 70 to 80%, not more than 80% in any case, right. Because there are so many different types of problems that are associated, okay, so many factors that actually governs the separation, right. And let us assume that we want a pure product. Or we want to separate or get rid of everything, all virus, not a single virus should be there in a therapeutic products. I want a 99.99% pure product. Okay. So, in that case what you can do? So, in biopharmaceutical industries where we are talking about bioseparation, protein separation, antibodies and all these things, usually after a batch concentration we feed them to the chromatographic columns to achieve 99.99% purity. So, that is the only way you can get so high purity, but you know chromatographic columns are very costly. So, due to this actually the downstream processing cost is becoming very high and the product cost becomes very high, right. To get rid of all these things, the membrane systems or membrane scientists have suggested that we can have cascade systems. Instead of having to do let us say we are doing the batch concentration of 60 initially 6 to 7% purity to 60 to 70% purity, then we are taking it to this one here chromatographic separation. Ultimately it has to pass through a chromatographic separation because no other separation will give you 99.99% purity, right. They are extremely selective. So, if we can have a series of cascades of membrane ultrafiltration systems, then we can actually concentrate it to a higher range than that doing it in more number of chromatography columns. So, you can operate multi-stage operations in different way. I am just trying to show one such things. Okay. So, let us see how it happens. Okay. So, this is our feed. Okay. Okay. See, this is unit 1, this is unit 2, this is unit 3. Let me draw, then I will explain. See, this is feed. Okay. So, we get permeate 1, we will get here permeate 2, we will get here permeate 3. Okay. So, we collect them all and we get overall permeate here. And here what is happening? Okay, the retentate from unit 1 is being fed as feed to the unit 2, right. Again the retentate from feed from unit 2 is the feed to the unit 3. So, what we get here? We get overall retentate here, R here. So, this is called in cascades, okay, or they are called in series. So, those who have read chemical reaction engineering, they must have read tank in series model, okay, where the equation of overall performance is usually given by eta t equals to eta 1 plus eta 2 into 1 minus eta 1, okay, plus eta 3, okay, into this total here plus it goes on. So, that was about our this one multi-stage operations, right. Now, we can have this multi-stage operations in a different way also. So, like I am just trying to just erase this, then we can I will just try to draw also some other small system here. Okay. So, let us say this is here. So, actually they are the same, but here let us say this gets divided. Here what is the difference between this system and this system? Here you see, initially you feed it, then the entire retentate from unit 1 goes as the feed to unit 2. Here it is not so. Okay. So, here the feed the retentate or R1 is divided and feed to 3 different units. Okay. Now, their retentate will be collected, okay, and again will be fed to cross flow systems, right. So, this is how it happens. Then the permeates will be collected, okay. So, this is the permeate, right, and this actually goes on. Now, let us understand the design of a batch cross flow system. So, let us assume that membrane has a constant rejection of the solute of interest. Okay. So, that means R equals to 1 minus Cp by Cb. That is equals to 1. So, Cp is your concentration of the solute in the permeate, Cb is the concentration of the solute in the bulk or feed, you can see. So, it follows that increase in solute concentration in feed tank from initial concentration Cb naught to the concentration at time t, that is Cbt, is proportional, okay, to the volume of solution remaining in the feed tank. So, this is the equation. So, Cbt by Cb naught equals to V naught by Vt. So, V naught is the initial volume of the tank, feed tank, and Vt is the volume of the feed tank at any time t. So, the volume of the permeate that is removed is nothing but V naught minus Vt. So, if the membrane is slightly permeable, that means R is less than 1, then the concentration ratio that can be achieved can be written in this equation. So, it is ln of Cbt by Cb naught equals to R ln V naught by Vt. So, you can see this particular diagram or the figure in which the concentration ratio Cb T by Cb naught is plotted against volume reduction, that is 1 minus Vt by V naught in percentage. You can see where R equals to 1, that means the constant rejection or the complete rejection, okay. You can see the volume that is reduced is the highest, right, and when membrane becomes semi permeable little, okay, your rejection reduces 0.8, 0.6, okay, you can see, right. So, the volume that is reduced actually getting less, right. So, with this we will wind up today's lecture. So, most of these lectures are taken from various books, and this one some from Becker, some from this one Mulder, okay. So, please refer to these books and read. In case you have any doubt, please feel free to ask me or write to me at kmohanty@iitg.ac.in, and in the next class we will discuss about the models of ultrafiltration transport such as pore flow model, concentration polarization model, mass transfer coefficient, membrane rejection and sieving coefficient. So, thank you very much.